Hello, dears, and welcome to Al-Hussaini Virtual Lab Pathology Talks, Tips, and Practical Tips. What I'm going to be sharing with you today is a nice case of a rare tumor occurring in the ovary, which is large cell in neuroendocrine carcinoma of the ovary. So this is a case of a 32-year-old female patient who presented with abdominal pain and was found to have an ovarian mass. And as you can see from the low power magnification, there is tendency for the tumor to grow in nests, in groups, in islands, separated by some edematous stroma, as well as a little bit of hemorrhage, and perhaps here some areas of necrosis. This appearance really should trigger the differential diagnosis of a neuroendocrine tumor. We should think about a neuroendocrine tumor once we see this very peculiar nesting or island type growth pattern in an ovarian tumor. Now, on high power magnification, as you can see, there is tendency for the tumor cells to grow like in sheets and then lots of mitotic figures. But on even further higher power magnification, the cells have this round vesicular nucleus, prominent nucleolus, and then an ample amount of acidophilic cytoplasm and plenty of necrosis and apoptosis in the background, and more of the same. The cells are large with vesicular nucleus, prominent nucleolus, and then large amount of what appears to be a granular acidophilic cytoplasm, an important clue to the diagnosis, and more of the same, another area showing those large cells with large abundant, with a large nucleus, prominent nucleolus, and then the abundant acidophilic cytoplasm. So the differential diagnosis really here, it contains or includes lots of entities. Germ cell tumors definitely enter into the differential diagnosis. Lymphomatous or hematolymphoid malignancies enter into the differential diagnosis, as well as different types of epithelial tumors, including neuroendocrine carcinoma. And this is the CAM 5.2 showing this diffuse strong positivity with tendency at this dot-like cytoplasmic staining. Excellent clue to the diagnosis of or potential diagnosis of a neuroendocrine carcinoma. What I like sometimes with immunohistochemical stains, that this counter stain with hematoxylin would really highlight some of the features like the extremely prominent nucleolus that we can see here, sometimes not very evident even at the H&E. So in addition, I always read the positive immunohistochemistry, but I always look into clues from the negative staining. So in the negative staining here, I can see the prominent nucleolus, and then with the positive stain, this strong positivity with dot-like cytoplasmic positivity. And then the cytokeratin NMF 116 showing the positivity again in some of the tumor cells, and again the dot-like positivity beautifully highlighting or supporting the possibility of a neuroendocrine carcinoma, and again a beautiful prominent nucleolus highlighted or seen sometimes vividly with the counter stain by immunohistochemistry. Now, one important potential pitfall is staining with CD138. 138, we know it as a marker for plasma cells in general and would support hematolymphoid malignancy. However, CD138 can be positive in a large number of carcinomas as well as even in glioblastomas. So we always have to interpret the immunohistochemistry in panels within the context of the case. So 138 does not negate the possibility of a carcinoma. On the contrary, it supports the possibility or the diagnosis of carcinoma in the right setup, in the right associated panel of immunostains. And then synaptophysin showing this dot-like cytoplasmic positivity beautifully seen in most of the tumor cells. Remember that sometimes with especially neuroendocrine markers, you have really to go on high power magnification to detect the positivity. And then the same with the Chromogranin-A, beautifully showing the dot-like positivity in most of the tumor cells. Remember that in order to render a confident diagnosis of a neuroendocrine carcinoma, you have to have two stains that are positive, and in this instance it was the synaptophysin and the chromogranin. Look at the beautiful staining cell here. Now, because of the initial differential diagnosis of possible germ cell tumor, a panel of immunohistochemical stains to rule out germ cell tumors, including the PLAB, OCT3/4, CD30, alpha-fetoprotein, and glypican-3 were all negative. And because of the possibility of hematolymphoid malignancy, CD20 and CD3 were performed and were completely negative. But remember, we have to put the rest of the immunohistochemical panels, the cytokeratins, the ones CD138, as well as the neuroendocrine markers together in order to render the diagnosis. Now, PAX8 was negative, thus ruling out most of the other differential diagnoses, inclusive of other types of carcinomas, especially poorly differentiated carcinomas or serous carcinoma in this example. Also remember that because the tumor shows an evidence of a neuroendocrine differentiation, we have to rule out other neuroendocrine tumors like the small cell carcinomas. And this is, as shown here, is the SMARCB1, the INI1, and the SMARCA4, which is the BRG1, show retained in nuclear staining, thus ruling out rhabdoid tumor or the small cell carcinoma hypercalcemic type of the ovary. So essentially, the final diagnosis of this case is large cell neuroendocrine carcinoma of the ovary. This is, although it shows a neuroendocrine differentiation, this is not a small cell carcinoma of the ovary because of the morphology, because of the larger nucleus, because of the prominent nucleolus. We also utilized immunohistochemistry to rule out other differential diagnoses like hematolymphoid, on different types of carcinomas, and like germ cell tumors. Now, large cell neuroendocrine carcinomas in general of the ovary or of the female genital tract are treated like other neuroendocrine carcinomas in the rest of the body. The patient received chemotherapy, appropriate chemotherapy, and there is evidence of a shrinkage of the mass in response as a feature of response to the treatment. One important also tip to remember that sometimes in neuroendocrine carcinomas of the ovaries in general, whether a small cell type or large cell carcinoma, neuroendocrine carcinoma can be associated with teratoma in the ovary. This is why it's very important to correlate with the radiological findings to ensure the absence of fat or bone in the ovary, thus helping to rule out the possibility of a teratoma. I hope you find this tip useful in your daily practice. Thank you.